Okay, good evening, everyone. So, in the last Saturday, if you quickly recall, we have started studying about the good evening, everyone. Prokaryotic transcription process. I hope every one of you remember it very clearly. If you remember the prokaryotic transcription, we have studied that and we understand very clearly that there is only one polymerase enzyme is there. There is only one RNA polymerase enzyme is involved, and that is only involved in the synthesis of three type of RNAs. Correct, na? Every one of you very quickly answer my question. Where is the site of synthesis of the tRNA molecule? Every one of you. Where is the site of the synthesis of the tRNA molecule? Everyone quickly. Where is the site of the synthesis of the tRNA molecule? No, Shanti, don't forget. rRNA is the only type of the RNA that is synthesized in the nucleoplasm. Whereas rRNA is the one which is synthesized in the nucleolus. tRNA and mRNA is the one which is synthesized in the nucleoplasm. Please don't get confused with that. Every student answer my question. In case of a eukaryotes, I am asking about eukaryotes, okay? In case of a eukaryotes, tRNA can be synthesized by what kind of a RNA polymerase enzyme? In case of a eukaryotes, the tRNA can be synthesized by which kind of a polymerase enzyme? Very good. Always remember my words. RNA polymerase 3 is the enzyme, friends, which is involved in the synthesis of all these small RNAs. Please kindly don't get confused with that particular point. Always remember that. Okay. Now, last and final question, friends. Including, I'm asking the word, listen to me, including the sigma factor, how many number of total subunits are present in the RNA polymerase? Including the sigma subunit, how many number of RNA? Very, very good. There are total 6. Alpha 2, beta, beta prime, omega, sigma. Please don't get confused with that particular point. Very good, very good. Okay. Now, we have also studied very, very clearly that, friends, if the RNA polymerase enzyme want to start that process of transcription, first of all, it has to come and attaches to the promoters. And we also studied very, very clearly that, friends, RNA polymerase enzyme never ever attaches to the promoters directly. If it wants to attach to the promoter, it requires the help of some protein. So what's the name of that particular protein that is helping here? That is only, friends, we call it as the sigma factor. Please kindly don't forget that. Okay, later, every student very quickly answer my question. Last and final question. What is the name of the enzyme that is most common in case of a replication and transcription? Everyone has to answer. What's the name of the enzyme that is very common in case of a transcription as well as in case of a replication? All of you. Very good. What was that enzyme? Topoisomerase enzyme. Please don't get confused with that particular point. So now, friends, what are the various stages in the transcription? Now you tell me. I don't tell you anything. I want every student has to answer. If all the students quickly recall, in the last class, we have discussed it till here. Now, friends, we are going to start the exact and original process. That is the process of initiation, the process of elongation, the process of termination. Okay, now you only answer my question. Every student has to answer. Here, before the initiation, I have written one more step. Recognition and binding. Every student answer. What has to be recognized and what has to be bind, friends? Every one of you, every one of you. What has to be recognized and what has to bind? Very good. RNA polymerase enzyme has to recognize the promoter. Then only RNA polymerase enzyme will come and bind it. Correct or not? Of course, yes. See, friends, if this is the promoter, without associating with the promoter, RNA polymerase enzyme never ever show any kind of a function. That every student commonly know about that. Okay. Now listen my statements very correctly without having a confusion. First step, recognition and binding. See, I am requesting every one of you, please try to understand from this particular slide. The more depth you understand, more easily you can answer the questions. Every student remember, who has to recognize? Promoter has to be recognized by the RNA polymerase enzyme. Look into the point, friends. RNA polymerase bind to its initiation sites through the base sequences known as promoters, which are recognized by the corresponding sigma factor. That you all people commonly know that. Now all the students look into the picture very correctly. Correct. Now listen, every one of you try to understand the concept correctly. Look into the point. See, friends, this is the DNA. See, every one of you has to recall this particular step, friends. It is very important. If every student quickly recall, in the last class, I told you, friends, sigma 2 will come and attaches to the -10 region. Understand the concept. Sigma 4 will attach us to the -35 region. There is no doubt about it. But friends, you know the point. Sigma 2 and sigma 4 attaches to the -10 and -35 very tightly. It's not at all loose attachment, friends. It's extremely tighter attachment. Now you only see the picture, what's happening here. Every student listen. So in the process of initiation, what's happening? See this binding of RNA polymerase. Look into this, friends. This is a RNA polymerase enzyme and this is a sigma factor. You got that point. Now sigma factor guiding the RNA polymerase enzyme. Look into this. It will come and attaches to the -10 and -35 very tightly. My friends, listen my statements very correctly. This is called closed complex formation. When do we call it as a closed complex? When it attached very tightly. But friends, listen my statements very correctly. This is what site? Every student follow me. This is what site? This is called INR. Initiation region. Friends, RNA polymerase enzyme has to come compulsory here. When it comes to the INR region, then only it will start the process of transcription. But what happened here? RNA polymerase enzyme is locked here because sigma factor tightly attached to the promoter. As a result, we call it as the closure complex. Now, what's the interesting point? Now, the question is very clear. How does closed complex can be converted into open complex? So most of the students will think that in order to convert the closure complex to the open complex, there is a requirement of the abundant amount of the energy. But all the students remember my words very correctly without confusion. If the closure complex wants to be converted into open complex, there is no requirement of energy. Look into the point. See, this is RNA polymerase enzyme, sigma factor. This is the promoter. Friends, just structural changes occur. What, friends? Structural change or conformational change. The structural or conformational change between what to what? Between the RNA polymerase enzyme and between the DNA. Because promoters are present on the DNA only, right? So friends, there is a simplified structural change or the conformational change occur. Because of that structural or conformational change, friends, what's going to happen? The closed complex converted into open complex. Friends, conversion of the closed complex to open complex, we call it as isomerization. What do we call? Isomerization. The transition, often called isomerization, does not require ATP. Then how does it happen? It just happened by the structural or conformational change. So friends, finally it is converted into the open complex. There is no doubt about it. Now all the students look into this diagram. This is the most important point all the students has to recall. Everyone remember, this is a RNA polymerase enzyme. Focus everyone. This is a sigma factor. So sigma factor guided RNA polymerase enzyme to come and attaches to the promoter. Initially, it form a closed complex. Later, closed complex under converted into open complex. You got that point correctly. Now, friends, tell me, every student understand my statements very correctly. See, now tell me, friends, this is RNA polymerase, right? Now, where it is present? It is present in the promoter. But friends, you know very well, in the last class, I told to all of you very clearly, from where the transcription will start. Transcription will always starts from the INR. What is INR? Initiation region. Every student keep the point in your mind. Whenever the RNA polymerase enzyme reaches the INR region, then only it will start synthesizing the RNA. Otherwise, it never start the synthesis of RNA completely. Once again, listen all the students. First, RNA polymerase enzyme comes and attaches to the promoter. After converting into open complex, RNA polymerase enzyme starts moving to the INR region. Why it start moving to the INR region? Because from there only it has to synthesize the RNA. But friends, what's the very interesting point here? RNA polymerase enzyme having a curious behavior. I'm telling you, what I'm going to tell you is, suppose example, your example I'm telling you, your examination is going to sharp at 9 a.m. Your examination is going to start at sharp 9 a.m. Some students will try to start the exam 8:45 a.m. only. But the app or the application never support you to start the exam. Same kind of a behavior RNA polymerase enzyme also trying to show. Before it reaching the INR region, it starts synthesizing the RNA in the form of a pieces. Small pieces. What do we call this kind of a initiation? We can call it abortive initiation, because this initiation never be continued. It will stop immediately. So remember, now the question is that why this particular initiation is happening? What is the reason? The reason is just because of the RNA polymerase enzyme. Every student follow my words. RNA polymerase enzyme having a curious behavior. Because of its curious behavior, before reaching the INR, it starts synthesizing small 10 nucleotides. But friends, now tell me, you can ask me, sir, what happens to that particular small 10 nucleotides? Friends, undoubtedly they get degraded because they are not stable. So friends, whenever finally all the students has to understand me, whenever the RNA polymerase enzyme has reached the INR, immediately what happened, friends? Look into the point. The sigma factor get dissociated. You can see that. The moment RNA polymerase enzyme reached, sigma factor get dissociated. And friends, remember my words. Now, once the sigma factor get dissociated, look into the point. RNA polymerase enzyme continuously synthesizing the RNA. This is what the process of initiation. Now, RNA is getting synthesized. Now, every student has to understand my statements very correctly. I'm only using the word prokaryotes. In case of a prokaryotes, friends, listen all the students. Transcription and translation occur simultaneously. What is the meaning of that? Listen all the students. Whenever RNA comes out, even at least 50 nucleotide of RNA comes out enough, immediately ribosome will come. You will see in the animation. And ribosome starts synthesizing the protein immediately, friends. It starts synthesizing the protein immediately. Why? Because in case of a prokaryotes, friends, transcription and translation are highly coupled. Why, friends? Because both are happening in the cytoplasm only. Please don't think that in case of a eukaryotes, transcription and translation are coupled. They never occur same time, because in eukaryotes, replication and transcription exclusively taking place in the nucleus. Never ever get confused with that particular point. Okay, now look into this. Now, if I ask you all a simple question to all of you, what happens in the elongation? Undoubtedly, we are synthesizing the mRNA. You only tell me, what is the meaning of synthesis of mRNA? Addition of one nucleotide with the another nucleotide. That's all. That is only we can call synthesis of mRNA. Every student answer my question. Now, if I want to synthesize the mRNA, every student know that mRNA is a single stranded. Only single strand. Now, out of these two strands, think and answer my question. Out of these two strands, which strand cannot be used by the RNA polymerase enzyme? My question to all of you is, among these two strands, which strand cannot be used by the RNA polymerase enzyme? Wonderful, wonderful. Coding strand or the non-template strand cannot be used by the RNA polymerase enzyme because friends, if sorry, if we want to synthesize the DNA, what do we require? We require only one strand, that is the template strand. Never ever get confused with that particular point. Okay, you understand that very correctly. Now, listen my words. See, friends, I hope you all the students remember mechanism of action is same, friends. Mechanism never ever changes. Always there is a fixed mechanism because addition of nucleotide is a common. So this is the first nucleotide having sugar, base, and here three phosphate groups are there. You know it. Same. Here second nucleotide, sugar, base, and another three phosphates are there. All the students look into this. This is the structure of a RNA polymerase enzyme. Both DNA polymerase and RNA polymerase enzyme structures are same, friends. If you look into the structure very clearly, RNA polymerase enzyme having two magnesium ions. You can see the picture associated with three aspartate residues. I have very clearly explained it to all of you in the DNA replication itself. Now you tell me what's happening. One magnesium ion will cut one phosphate group. One magnesium ion will cut one phosphate group. Another magnesium ion is doing what? Tell me. It is releasing the two phosphate groups in the form of a PPI. As a result, friends, two nucleotides are added. That's such a simple step it is. Two nucleotides are added. Correct, na? Now all the students try to understand me very correctly. Focus what exactly I'm saying here. Now what is going to happen? RNA polymerase will move here. When the RNA polymerase enzyme moved here, what's happening once again? Once again, friends, it will add another nucleotide here. Again, it will move. Again, it will be keep on adding. Like that, RNA polymerase enzyme continuously adding one nucleotide with another and it synthesizes the mRNA. Now, friends, listen, all the students understand. In order to explain how exactly RNA polymerase enzyme synthesizes the mRNA, in order to explain this process, there were two models were proposed. Now you people has to tell me which model is idealized model, which model is an accurate model. Now all the students look into the diagram very carefully and try to understand what exactly I'm saying to all of you. According to the first model, this is the first model. Listen, all the students. This is what, friends? DNA. Correct, na? Everyone know, right? This is how the double-stranded DNA look like. My friends, you know very well. This is called as a minor groove, major groove, minor groove, major groove, minor groove, major groove. You know what first model is saying, friends? Whenever there is a synthesis of RNA, RNA polymerase enzyme also will move to the major groove, minor groove, to the major groove, minor groove, major groove, and minor groove of the DNA. So, friends, whenever RNA got synthesized, now you tell me, friends, how can you separate this RNA? Do you think it's very easy? No. If you want to separate this RNA again from the DNA, we require a lot of enzymes. Then only we can separate this. Now, why the RNA got synthesized like this? Because RNA polymerase also moving towards the major and minor groove of the DNA. So however it is moving, RNA is also synthesizing in that way. So it's a very big headache. Again, we require a lot of enzymes. Now, if you look into the second model, according to the second model, all the students can see the diagram very clearly. Out of two strands of the DNA, RNA polymerase enzyme is using one strand of the DNA as the template and synthesizing the mRNA continuously. But the point is very clear. Whenever the mRNA getting synthesized by the RNA polymerase enzyme, here the coiling occur. What happens if the coiling occur? No problem, right? We have a topoisomerase enzyme. It will come and remove the coiling. But friends, at the end, there will be a no problem to disassociate or to remove this RNA from the DNA. It's very, very easy. There is no need to get no need to use any kind of enzyme. Why? RNA polymerase enzyme is moving along the double-stranded DNA and taking one strand as the template and synthesizing the mRNA. Now, my question is, among the first and second model, which model can be widely used by the RNA polymerase enzyme in order to synthesize the mRNA? Undoubtedly, we don't need to think it. Undoubtedly, second model. Why? Because in the second model, the enzyme is also moving along with the double-stranded DNA only. There is no doubt about that. Correct, na? So now the point is very, very clear that even though it is moving like this, all the students try to understand the point correctly. Even though it is moving like this, coiling occur, but there's no problem. We have a topoisomerase enzyme. That's why it always move the second model. Please don't get confused with that particular point. Then friends, most important, you will get a direct question from the termination itself. It's also very, very important, friends, termination. And the termination in case of a transcription is very different compared to the replication. All the students listen, look into the diagram, every one of you. This is the double-stranded DNA. This is a RNA polymerase enzyme. RNA polymerase enzyme is moving continuously and synthesizing the RNA. You can understand. Now, my question is that till how long RNA polymerase enzyme keep on moving on the double-stranded DNA? That is the question. How long it will move? RNA polymerase enzyme is keep on moving the DNA until it reaches a particular site. What is that particular site? That is called termination site. Tell me, Haimawati, termination site is present on the DNA or RNA? Every student answered. Termination site is present in the DNA or RNA? Every one of you. Undoubtedly, termination site is present on the DNA. Correct, na? The RNA not only synthesized, how does the RNA having the termination site? It is only present on the DNA. Now look into this. Look into this. See, I'll explain you briefly and elaboratively to all of you. Focus. There are two types of terminations are possible in case of a prokaryotic organism. What are those, friends? One is called as spontaneous termination. There are many names are there, friends. The spontaneous termination, also called as intrinsic termination. What do we call it as? Intrinsic termination. Then the next one we call it as, every student remember my words correctly, extrinsic termination or non-spontaneous termination. Now you have to understand on your own. Why did I use the word spontaneous or intrinsic? Which means that, friends, the mRNA coming out on its own. No one is taking the mRNA out from the DNA. On its own, it will come out. That's why it is called intrinsic or spontaneous. But friends, when the mRNA is not coming out on its own, it requires the help of certain external enzyme, external protein. Whenever it take the help of an external protein or an external enzyme, that is called extrinsic termination. That is called non-spontaneous termination. They can ask the question in the exam. What kind of termination is most commonly used by the mRNA in the process of termination? Undoubtedly extrinsic, because friends, every single time mRNA never comes out on its own. That is not possible. So that's why it requires the help of many proteins. That's why we can call it extrinsic or non-spontaneous termination is the most common type of the termination. Never ever get confused with that particular point. Okay, now look into the picture correctly, all the students. Okay, there are two types of termination, extrinsic, intrinsic, spontaneous or non-spontaneous. I hope you remember, right? Extrinsic means what I told you. It has to use some protein. You know what is the name of that protein? Rho protein. What is called as rho protein. So that's the reason, friends. Extrinsic termination, also called as rho dependent termination. Simple point it is. Spontaneous or intrinsic termination, also called as, all the students remember, also called as what we can call row independent termination, which means it is not at all using the row protein. Without row protein, it is coming on its own. Look into that. How is that possible? Now all the students listen, how nicely RNA is coming on its own without taking anyone help. So look into this. I have called it as row independent termination. Why did I use the word row independent, friends? It's not using anyone help. Look into the point. Look into the point. This is the DNA. You can see, friends, what is coming out. This is the mRNA that is coming out. Every student focus on the diagram very clearly. You can see one thing. Still, friends, look into this. Still the mRNA, listen, still the mRNA is attached with the DNA only. You can see, right? We have to bring it out. How is that possible? RNA only bring it out. How? Listen, all the students. So this is the mRNA, right? All the students remember. First 50 to 60 nucleotide of mRNA, listen every one of you, it contain more number of guanine and more number of cytosine. What, friends? First 50 to 60 nucleotide of mRNA contain more guanine and cytosine. We all people know very well, according to the Watson and Crick, guanine always pair with the cytosine with the 3 hydrogen bonds. Now all the students look into the picture here. Here there is a more number of guanine here. You can see that. And here there is a more number of cytosine also. Correct, na? There's no doubt about it. Now the question is very, very clear, friends. How does the guanine is going to pair with the cytosine? With the help of hydrogen bonds. Now you look into this. Whenever guanine pair with the cytosine, see this? It is forming a hairpin loop-like structure. Correct, na? So the ladies will keep a hairpin. So when the guanine and cytosine pair, it is also exactly look like a hairpin. So now you can see that. Now the point is very clear. Now the hairpin loop will provide the strength. What, friends? Remember my words. The hairpin loop will provide the strength to pull this RNA. Listen my statements correctly. The hairpin loop will pull the mRNA in such a way that it will break down from the DNA. RNA polymerase will be dissociated, and some students will ask a very good doubt. Sir, then what happens to the hairpin loop? Friends, actually, it is pulling the mRNA very strongly, right? Because of that force, hairpin loop also will become linear. So what I'm trying to explain you, listen all the students once again. The first 50 to 60 nucleotide of mRNA contain abundant amount of guanine and cytosine. And guanine and cytosine paired with each other with the help of a hairpin loop-like structure. When they form a hairpin loop-like structure, what you have to remember, friends, the point is crystal clear. Listen to me correctly. Now, friends, this hairpin loop-like structure provide the strength, provide the force to pull. See, friends, it's a simple like, this is a thread. By using the thread, we are pulling a vehicle. So here, what is the vehicle? Vehicle only RNA polymerase plus DNA. Correct, na? If I pull it a great pressure, sometimes what happens? Thread will break down. So this will get separated. Same thing will happen here. The hairpin loop will provide the strength. By that, RNA polymerase enzyme will dissociate, RNA gets separated. So this is called row independent. Why? Because we have not used any external protein, any external enzyme. So it became row independent. But this is not possible all the time. Why? What's the reason? Why it's not possible all the time? The reason is that every single time hairpin loop is not sufficient. Suppose example, friends, I want to pull a vehicle. The vehicle is too much weight. Now I am the only person pulling the vehicle. Do you think it is possible for me to pull this large vehicle? It is not sufficient. Same thing. Sometimes hairpin loop is not stronger to pull the RNA. Then it compulsory require the help, right? So I will take the help of a second person. I will take the help of a third person. Same way, every time hairpin loop is not stronger, they require the help of a external protein called as row protein. Never ever get confused with that particular point. So friends, whenever there is a use of that external protein or an external enzyme, that is called as row dependent termination. Now you see this point, what I have written. Every time it is not possible to form strong hairpins. Every student remember, every time it is not possible to form a strong hairpins, then my friends, we require an external protein named as Rho protein. And friends, very important question for the exam. The Rho protein belongs to which family? This is an important question. The Rho protein will belongs to a family called as Rec-A family. Friends, please don't get confused with that. So what was that? Rho protein is a Rec-A family hexameric helicase having 6 subunits. Now you only look into this. What's happening here? Such a simple statement it is. Now you all people has to ask me one more good question. Sir, this is mRNA. That is a good question. Here you can see, right, 6 subunits, right? That is only called Rho protein. This entire thing is called Rho protein. Now you have to ask me a question. Sir, where exactly the Rho protein will come and attaches to the mRNA? That's a valid question. Where exactly Rho protein come and attaches to the mRNA? See friends, on the mRNA, there are certain sequences are present. We can call the RUT sequence, R-U-T. Rho utilization terminus. I want you to remember as a RUT. Okay, Rho utilization terminus. My friends, now the Rho protein will come and attaches to the RUT sequences. Such a simple point it is. Whenever Rho protein comes and attaches to the RUT sequence, now you listen what's happening. Now you know what the Rho protein is doing. Rho protein is moving till the hairpin loop. See here, already hairpin loop is formed, right? So Rho protein will move till the hairpin loop. Then it will provide extra support. It will provide the extra support to the hairpin loop. Then it can pull the mRNA very easily from the DNA. You can see that point. As a result, it can easily separate. Never ever get confused with that particular point. So this is what Rho dependent termination. Okay, just give me one moment of time. I'll show you a simplified animation. Then you'll get a little more clear-cut idea about this. Just give me one moment of time. Okay, every one of you, please look into this. So simplified animation it is. All the students pay attention. Can you hear my voice? Am I audible? Quickly tell me. Yeah, am I audible? I think I am not audible. Okay, that's good. Okay, continue. So all the students look into this. This is a prokaryotic cell such as the bacteria. Look into that. I want you to see it very correctly. So that is the DNA. Now you all people can see the promoters, friends. You can see, right? -10 region, one promoter, Tata box. -35 region, there is another promoter. You know that. Now that is what? RNA polymerase core enzyme, sigma factor. Together call it as what enzyme, friends? Holo enzyme. Now where it comes and attaches, tell me. It attaches to the promoter. I told to all of you, right? Friends, this is called a closed complex formation. Now you see that closed complex converted into open. See that? So it just converted into open. How is that possible? Just by the structural change or the conformational change. We do not require ATP at all. Then where it will move, friends? It will move to the INR region by using one strand of the DNA as the template. Now sigma factor will be dissociated because it is not required, right? So see that. Now it reached INR. Now it will start the process of transcription. Now you see that. See that nucleotides are adding continuously. What are those? mRNA. Now this is the initiation. Now the most important you have to remember is the elongation. Hope every student remember that I have told to all of you, whenever the mRNA is getting synthesized, protein synthesis also will start immediately. Why? Because in the prokaryotic organism, the process of a transcription and the process of translation is a highly coupled process. Look into that. Look into that. See that mRNA is coming out. Now what will come? Look into that. Ribosome is coming now. See that ribosome came there and it starts synthesizing the protein directly. See that? Now you can see, right, the small green color one is coming out. That is only protein. Now, once the RNA polymerase reaches the termination site, two type of terminations are happening. Row dependent, row independent. Look into that. Look into that. This is what, friends? Row dependent termination. You can see that point. Row dependent termination. Now, what you have to understand, there is no requirement of anyone help. See that, see that. Hairpin loop got formed and it is only pulling it. See that? It will only pull it. See this, see that. Now it automatically become linear also. RNA polymerase enzyme dissociated also. But what's the point? The point is that it is not possible all the time. Why? Because hairpin loop is not stronger all the time. So what we have to do? Now we have to bring extra protein. Now you see that. This is the row dependent. Now who will come and attach us? Undoubtedly, see that row protein will come and attach us. Now see this. Row protein is moving. Where it is moving? Till the hairpin loop. So look into that. Look into that. It moved till the hairpin loop. You can see that. Now it gives an extra support for the hairpin loop to pull the mRNA. See that. So it's pulling it now by giving an extra support. It become linear, right? So that is about row dependent and row independent termination in the prokaryotes. Undoubtedly, friends, in every organism, prokaryotic transcription, prokaryotic replication, prokaryotic translation is easy compared to the eukaryotes. Why? Because we are very complex organism. Our genome is a very large genome. There is a requirement of lot of proteins. There is a requirement of lot of enzymes. But the point is that without understanding prokaryotes, it is extremely difficult to understand the eukaryotes. Now let's focus on the eukaryotes. Okay. Everyone quickly answer. Is it clear, everyone? Prokaryotic transcription, quickly confirm me. Is it clear for everyone? Prokaryotic transcription faster. Don't take too much of time. Okay, that's perfect. So now what we have to understand is the eukaryotic transcription. Everyone quickly answer my question. In case of a eukaryotic transcription, where the transcription is taking place? Where is the site of the transcription in the eukaryotes? All the students. Undoubtedly, it is taking place in the within the nucleus only, because all the RNA polymerase enzymes are synthesized within the nucleus only. Friends, please don't get confused with that particular point. Now, all of you pay attention. RNA polymerase 1, what it is synthesizing? rRNA in the nucleoli or nucleolus, anything is same. RNA pol2 transcribe mRNA, pol3 transcribe all the type of small RNAs in the nucleoplasm. All the students, please listen my statements very correctly without having a confusion. This is also a question that they will ask in the exam, and this is also a question which is asked previously. What's the question? There is a name of a fungal toxin. Okay, there is a name of the toxin which is derived from the fungus named as alpha amanitin. Alpha amanitin is the name of the toxin which is isolated from the fungus or released from the fungus, which will inhibit polymerase enzymes. Now, what is the question they asked in the exam? Arrange the following RNA polymerases according to the sensitivity towards the alpha amanitin. Either in the ascending order they can ask, or in the descending order also they can ask. No need to get confused with that particular point. Now, all the students try to understand my statements very correctly without having a confusion. RNA polymerase 2. RNA polymerase 2. It is the one which is transcribing mRNA, right? Major enzyme. this will show more sensitivity, which means that, friends, I can say in a simple lines to all of you, alpha amanitin can easily destruct this enzyme. And RNA polymerase 3, it will show intermediate sensitivity. Only 50% of the RNA polymerase 3 inhibited by the alpha amanitin. What about the polymerase 1? Polymerase 1 never show any sensitivity, which means, friends, alpha amanitin cannot destruct this particular alpha amanitin cannot destruct pol1. It majorly destruct pol2 and very less percent it destruct the pol3. So remember, according to the sensitivity wise, pol2 followed by pol3 followed by pol1. Always remember this. Don't get confused with that. Okay. Now I hope you all people remember similar kind of a diagram we all people have studied in the prokaryotic transcription also. Okay. Just give me one moment. Okay, now listen, all the students listen, look into the point. Same, suppose this is the RNA polymerase enzyme. You do the cross section. When you do the cross section of an RNA polymerase enzyme, look into the point. Same, many channels you can observe. You can see, right? One is called downstream channel, correct na? See this is called exit channel. What is the meaning of exit channel? Through which RNA goes out, correct na? One is called template strand, another one is called as the non-template strand. Now the question is very, very clear. You know, friends, in order to synthesize the mRNA, what strand we require? We require only template strand. Let's consider this blue color one is the template strand and red color is the RNA which is going out. Friends, who will hold or who will attach to the, what I can say, mRNA and template strand? You can see, right? Clamp and rudder. What was that? Clamp and rudder. It's like a rubber band. It's like a rubber band which hold two things together. It will hold the template strand of the DNA. It will also hold the mRNA also very tightly with each other. Don't get confused with that particular point. Okay. Now, from now onwards, the lesson will become very difficult. It's not difficult, very much confusing, because lot of proteins are involved. If you don't understand properly, you can't answer the questions. Now, in case of prokaryotic organism, we all people studied that RNA polymerase enzyme using only two type of a promoter. One promoter present at -10 region, another promoter present at the -35 region. You understand that concept very correctly without confusion. Now, all the students listen to me correctly. In case of eukaryotes, there are many promoters are there, not one or two, friends, many are there. Now, first of all, why I have written the word class 2 promoter? Why I have written like this? Because we are talking about RNA polymerase 2. Why we are talking about RNA polymerase 2? Because our ultimate goal is to synthesize mRNA. That is our ultimate goal. So that's why did I use the word class 2? Because RNA polymerase 2 only recognize these promoters. That's why we can call it class 2 promoters. Please don't get confused with that point. Every student quickly answer. Suppose this is the INR, initiation region. In case of a prokaryotic organism, the promoters are present towards the left-hand side or the promoters are present towards the right-hand side. I mean to say, is it a upstream promoter or is it a downstream promoter? Every student, I'm talking about in prokaryotes. I'm talking about prokaryotes. Is it a upstream or is it a downstream? Undoubtedly, friends, it is upstream only. But all the students remember my words very correctly without having any confusion. In case of a eukaryotic organism, all the students has to follow me. In case of a eukaryotic organism, there are two type of promoters are there. One is called core promoter, another one is called proximal promoter. Every student remember, core promoters, suppose friends listen, suppose every one of you listen, this is a INR. This is a INR. Core promoters present in the upstream also, present in the downstream also. Right. And what about the proximal promoters? Proximal promoters are majorly present in the upstream only. Now, the question is very, very clear. Now, RNA polymerase 2 enzyme is using what kind of a promoters? Undoubtedly, friends, RNA polymerase enzyme is using a proximal promoter because it has to synthesize the mRNA, which is present on the upstream. Now, all the students listen. So see this, how many promoters are there? Core promoter, proximal promoter. Is it necessary to remember all these? It is mandatory to remember the names. I don't want any one of you to remember the sequences. It's not required. But I am requesting every one of you to remember the names compulsory. Okay. Listen everyone. What is the first promoter, friends? Core promoter. Correct na? What I told you. Core promoters present in the... See, friends, it's very tough for the first time. When you read 2 to 3 times only, you will understand. Okay. Now, upstream and downstream, both areas core promoters are present. From where? From the INR. Every student look into the diagram correctly. This is the INR. From the INR, there are 2 core promoters are present upstream. There are 3 core promoters are present on the downstream. You can see that. I don't encourage any one of you to remember the sequences. I encourage all of you to remember the names, that's all. From the INR towards the left-hand direction, there is a 2 promoters. One is called Tata box. Another one is called BRE promoter. I want you to remember the short names all the time. You won't get the question from the full forms. Now, on the downstream or towards the right-hand side, you can see, right, friends, there are 3 promoters are present. Listen, everyone. We call it downstream core elements. See this? Which are in the yellow color. We call it them as the downstream core element. We can call DCE. And blue color one you can see, right? That is called MTE, motif 10 element. It is called DPE, downstream promoter element. Please don't worry about that. You never get in the exam. I'm just telling you for your better understanding. From the INR, there are 2 core promoters are present in the upstream, and there are 3 core promoters are present on the downstream, named as DCE, MTE, DPE. You got that? That is only core promoter. But what is the other promoter? Most important promoter, proximal promoter. Where the proximal promoters are present, every student listen. This is the INR. From the INR, every student remember, 70 to 75 nucleotides. Minus 72, minus 75 nucleotides. There is a promoter is present. That promoter is rich of G and C sequences. Same from the INR at minus 90 nucleotides. So if you count 1, 2, 3, 4, 5, 6, 7 like that, at minus 70 to 75, GC boxes are present. Means GC boxes means they are rich of guanine cytosine. Guanine cytosine. What about minus 90? At minus 90, there is a repeated sequence of CAAT. That's why this box is called cat box. Why? Because there is a repeated sequence of CAAT sequence. I don't encourage you to remember it. They won't ask in the exam. Please don't worry about it. But you have to understand. Without understanding this, you can't get the concept. Now, before entering into the concept, all the students has to remember my statements very correctly. What are enhancers? What are silencers? Friends, name itself indicating, right? Enhancers, which enhance the process of transcription. Silencers, which silence or which suppress the process of transcription. That is what exactly we can call silencers. Now, the question is very clear. How they enhance the transcription? How they suppress the transcription? All the students look into the point very correctly. Look into this beautiful picture. Look into that. See, friends, every one of you listen my statements. Don't get confused here. Listen, just try to understand. Suppose example, this is the promoter, right? Correct, na? This is the promoter where the RNA polymerase enzyme will present. Correct, na? Here RNA polymerase enzyme will come and attaches. But friends, you know where the enhancers are present. You see this? Enhancers are present here. This is enhancer. Friends, you understand. Enhancer is located very far from the promoter. Now the question is very clear. If the enhancer is located very far from the promoter, how they can enhance the transcription? That's a good question. With the help of, you can see that, with the help of a transcription factors and mediator proteins, you can see all these. So my point is, it doesn't matter how far the enhancer may be situated. It doesn't matter how far the enhancers are arranged. They can enhance the process of transcription compulsory by using transcription factors and mediator proteins. Now, what is opposite? Tell me. Silencers. I'll tell you in a simple words to all of you what the silencer will do. Simple, friends. All the students listen. Ultimate goal of the silencer is what, friends? Ultimate goal of the silencer is suppress the transcription, which means, friends, it should not allow the process of transcription to occur. So, you know, this is the DNA, right? What the silencer will do, example, silencer will attach us to the DNA here. You know what it will do? It will cause the DNA to undergo coiling. You can see that. Compact structure, condensed structure. In simple words, I can tell to all of you friends, silencer will convert the DNA into heterochromatin form, which is a, what I can say, which is a condensed or compact structure. Now you tell me now, this is a RNA polymerase enzyme. All the students answer me now. Do you think RNA polymerase comes to know where is the promoter now? Can it recognize? Can it recognize where is the promoter exactly? No, it is not possible. Why? Because it is completely compacted. It is completely condensed. RNA polymerase cannot recognize the promoter. If the RNA polymerase is not recognizing the promoter, how does the process of transcription occur? Never. It will suppress or it will stop the process of transcription. Such a simple statement it is. So what I'm trying to explain you, friends, enhancers enhance the transcription with the help of transcription factor and mediator proteins. Silencers will condense the DNA into heterochromatin form and it suppress the transcription. Please don't get confused with that. So now we have to understand how does the process of transcription takes place. Now we are going to understand the process of initiation, elongation, and termination. It is not at all simple like prokaryotes. Prokaryotes, it's very, very easy. It's very difficult. Now all the students remember, they can ask a question directly in the exam. What question they can ask in the exam? Which of the following proteins will control all the transcription process? In other words, which of the following proteins will control initiation, elongation, and termination in the eukaryotic transcription? Who will control? Transcription factors will control. They'll control all the 3 steps. We will discuss in the tomorrow class elaboratively, but remember, there are 2 types of transcription factors: general transcription factors, promoter specific transcription factor. I can tell to all the students in a very simple words, whenever I use the word general transcription factors, every student remember, by using these transcription factors, transcription occur. There is no doubt about it. Transcription occur. But the transcription occur at a basal level. The transcription is not 100%. The transcription is not efficient transcription. So, but what about the promoter specific transcription factor? They attach us exactly at a specific sequences and they will allow the transcription to occur at the maximum level. Please don't get confused with that particular point, which we can call promoter specific transcription factors. In the tomorrow class, we will understand how the initiation, elongation, and termination occur. Okay. Clear, everyone? I think it will take another 5 days. Okay. Either 9th or 10th, it will be done. Clear, everyone? Okay. Okay. Perfect. 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 Okay. That's all for now. I'll see you in the tomorrow morning class. Okay. 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 Thank you. Thank you all. Thank you. Please Please revise whatever the questions that I have given. Please practice. Don't neglect under any circumstances. Keep practicing the questions. Okay. Thank you.